Welcome to another the eBiology Teacher Podcast. I am Mr. Doc, and today we're talking about DNA replication and how DNA is able to be reproduced or replicated so that cells can divide. So today we're going to talk about the process. We're going to talk about the roles that enzymes play. I know we haven't talked about enzymes since we talked about the digestive system. We're going to explain the importance of complementary base pairs. Yes, there's a reason you learned that C's match with A's, or T's match with A's, and A's match with T's, and so forth on. And then we're going to talk about what will happen if there's an error in the DNA replication process. And so Ben is standing over here trying to distract me. That's how I'm trying to do this.
Doc.
Go away.
Don't
go
mess
away.
up, Doc.
All right. So let's go ahead and get started. First, let's preview. We're going to talk about mitosis later, but there is something that we do need to understand. There are two major phases in a cell's life cycle. First is interphase, and the second is a cellular division or mitosis. In interphase, which is basically the life of a cell when it's not replicating, there are three subphases. G1, which is the growth phase, where it's just been produced. It's either just reproduced or have been produced. The S phase, where the DNA is copied, and the G2 phase, where the cell matures to the point where it can actually mature enough to reproduce. In the S phase of interphase, the cell's DNA reproduces. This is important because without this phase, a cell can't pass on its genetic information. The genetic information is what tells the cell what to do. So if it gives away its instruction manual, then it can't go on living. Right? So what has to happen is that the genetic information has to be copied so that the cell can keep one copy for itself and give a copy to another cell. Now, the next thing we're going to look at, sorry, I'm doing something new today and it's really not working out for me. There we go. DNA replication is the process by which DNA is copied. Enzymes will unravel or untwist the DNA molecule and unzip it, exposing the nitrogenous bases. The nitrogenous bases are what make up the code that DNA is written in. All right? So it's like a sentence. The process ultimately creates two DNA strands, each strand having half the old strand and half of a new strand. So let's take a look at the process. An enzyme called helicase will literally unzip the DNA molecule. What that means is, if you look at the screen here, the helicase molecule, which is located right here, this little greenish yellow thing, it unzips it like a zipper. It's like the pull on a zipper. When it does that, the DNA nitrogenous bases are exposed. PJ just walked in and they won the choir competition. Congratulations.
Yeah.
Grand champions.
Yeah.
Our own.
Two in a row.
That's it. Counterpoints rock and roll.
We got to stay in 2 weeks.
Good luck. Sorry for that. It's a passing period right now. It's probably not the best time to record this. Anyway, the helicase opens up what's called the replication fork. In other words, we have two half strands of DNA that are exposed. The nitrogenous bases are there, ready to be copied. The purple little things here are called DNA polymerase. Polymerase, if we translate it, means a polymer builder, something that makes something. A polymerase attaches one to each strand, and then it's going to be able to copy those strands. We do have a bit of a hiccup here, though. Remember we talked about the direction of DNA, 5' and 3' and so forth. Well, that's where the importance of this comes in. DNA polymerases travel up the strands from a 5' to 3' direction. Much like we read from left to right, DNA polymerase leads from 5' to 3'. That's 5' to 3'. Here's the problem. If we look at a DNA strand, there's one that goes 5' to 3', and then there's one that goes 3' to 5'. So one's kind of upside down. The one that's right side up, or runs from 5 to 3, is called the leading strand. And the one that's from 3 to 5 is called the lagging strand. Now here's what happens. The leading strand, let's say right here in this figure, the one where you see the little white hand, that is 5', and this is 3'. What happens is this DNA polymerase simply goes and just adds on the new nitrogenous bases, making a new strand. That is 5' to 3'. But remember, this one is 5' over here and 3' over here. So it has to replicate in that direction. Whoop. Here's the problem. the opening end of that DNA strand is it hasn't been unzipped yet. So it can't just start copying. It's going to have to copy in little fragments. So here's what happens. The polymerase goes and it makes a little fragment, copies, jumps off, attaches here, copies, jumps off, attaches here, copies, jumps off, attaches here, and copies. So the leading strand works like a computer. You just type, just keep going, and it just keeps going and keeps going, and it knows where to cut and stop and all that other stuff. The lagging strand's more like a typewriter. You go type, ding, type, ding, type, and so forth on. So it's doing it in little segments, okay? And that's how we get around that. Now, once this is done, we have 2 DNA strands because the DNA polymerase will just fall off and we're ready to go, and the DNA will naturally go back into its helical shape. So that's how it works. Let's take a look now at the result. First of all, we have 2 DNA out molecules that are identical to one another. That's because we started off with the same template. But here's the other thing. Each strand is composed of half of a strand of the original molecule and a strand of new DNA. So in the double helix, the one where we were starting with a leading strand, that leading strand is part of the original DNA molecule, and then the new DNA is formed on the other side, creating a new lagging strand. The lagging strand from the original DNA molecule remains in that piece of DNA with a new half on the other side. This is called semi-conservative replication because original part of the DNA that we started with is in each of the 2 new molecules. All right? Now let's take a look at a movie, and I want you to watch what happens. Now, first off in this diagram, notice that the original molecule has purple bands, okay? We're just going to call those purple strands because it makes it easier to see. Then we're going to watch what happens, okay? So let's take a look.
Replication begins when enzymes called helicases unwind the DNA into 2 separate strands. Next, enzymes called DNA polymerases move along the separated chains, assembling new chains using complementary
Notice that the new
nucleotides.
DNA is in blue.
The result of replication is 2 identical DNA
2
molecules, each made up of an original strand and a
Half
new strand.
purple, half blue, right? That's the way it works. Watch it one more time. This time I want you to look at where the
DNA
leading strand
replication
and lagging
begins
strand
when
are.
enzymes called helicases unwind the DNA into 2 separate strands.
Watch
Next,
this.
enzymes called
This one is the
DNA
leading strand because it's just
polymerases
going
move
up,
along the
This
separated
one is the lagging
chains,
strand because it starts up
assembling
here
new
and works its way
chains
down,
using
starts up here and works
complementary
its way down, starts up here and
nucleotides.
works its way down, starts up here and works its way down. So the one on
The
the
result
left
of replication
is
is 2
strand.
identical DNA molecules, each made up of an original strand and a new strand.
All right. Now that we understand that, what happens if copying or replication goes wrong? Well, that results in mutations, and a mutation is simply a change in the DNA sequence. We hear about mutations and we instantly think, wow, bad news, mutations are bad. They can be, that's true, but the overwhelming majority of mutations not only aren't harmful, they go unnoticed entirely. We never know that they're there. That's the majority of mutations. Some of them can even be beneficial. So you can't just say that all mutations are bad or mutation's a bad thing. As a matter of fact, sometimes mutation has been incredibly beneficial to many, many people. Mutations can be described in 2 ways. First of all, there's a point mutation, and it's kind of hard to see, I know, because it's kind of small, but in this mutation, in a point mutation, a single nucleotide has been substituted with another. So in this case, the sequence should be G, G, C, but one of the G's has been replaced with an A, creating an A, G, C codon, or a period of 3 nucleotides. What happens here is instead of a glycine being added when we make, say, protein, a serine is added. Well, that means that we have changed the structure and therefore we have changed the function. There you go. Now, a frame shift mutation is a mutation that results from the insertion or deletion of a nitrogenous base or nucleotide. And not only does it affect like a point mutation does, but it affects everything after it. So here, in opposed to changing one of the G's to an A, we've just inserted a new A, right? Well, what happens here is that this A shifts everything down one, which means that everything that comes after this mutation is going to be wrong. So in a point mutation, we have like a single amino acid that's affected. In a frame shift mutation, everything from the point of the mutation on is affected. So it's obviously a little more broad. What causes mutations can be a variety of things. First of all, sometimes there's just a mistake in the copying and replication process, much like a typo. Radiation can knock DNA out of sequence, which is why people who work around radiation wear lead vests. And if you've ever been to the dentist office and they lay that lead apron over you, it's to prevent any sort of hazards from the radiation. Chemicals called mutagens are chemical agents that can cause mutations. A specific type is called carcinogens. Carcinogens are a type of mutagens that are known to cause cancer. Viruses can introduce new genetic information. For example, in the 60s, there was an outbreak of the German measles in Indiana. Shortly thereafter, the number of babies born that were deaf skyrocketed by about 80%. Turns out that the measles, the German measles, can mutate the DNA in sex cells, sperm and egg, and therefore the offspring were born deaf. Scientists can even do what's called directed mutation, in which they intentionally change DNA. The prime example of that I can think of, though it is fictional, is that of Jurassic Park. They took frog DNA and inserted some DNA that they got from an insect preserved in amber, and voila, you've got a dinosaur. Yes, so we can do that. Many of the foods we eat have been directly mutated. For example, there have been tomatoes that have undergone mutation to let's say, stay on the shelf longer so they don't rot before they get to market. Or chickens have been genetically enhanced to produce more muscle or stronger bones, whatever. Those are mutations that occur by direction of those scientists who are trying to attempt to do something. The resultant mutations of DNA can result in a number of things. It can go undetected, as we've already mentioned. We never notice it. It can also change the function of a cell, decrease or increase. Some mutations can lead to an increased uncontrolled division of cells, which is called cancer. Other mutations can lead to a production of beneficial traits. There's a family in northern Italy who have eaten fried sausages and all kinds of horribly unhealthy things for them, and none of them have heart problems. None of them have cholesterol problems. It's because they have a genetic mutation in their family that prevents them from collecting fat in their blood vessels and plaque. Since the DNA is replicated and cells divide, it is possible to send mutations to the offspring if and only if the mutation occurs in the gametes being either the sperm or the egg. For example, the German measles, which we already talked about, obviously mutated the sex cells. It did not necessarily mutate the body cells. So it was able to be passed on to an offspring. But let's say we get radiation poisoning and you get some type of cancer from radiation. If that radiation did not affect the sperm or the egg, that will not be passed on to the child because the DNA in the body cells is determined by what was in the parent's sex cells. If it's not in the sex cells, it's not going to be passed on. Another example would be that of scarlet fever. If scarlet fever goes untreated, it can lead to blindness. But if someone becomes blind because of scarlet fever, that's not something that's going to be passed on to the offspring because it doesn't affect the gametes DNA. It only affects the somatic cells DNA, somatic being body cells. The last thing I want to talk about is the reality of superheroes. I have a lot of questions sometimes. Is it possible for somebody to whatever? All right? It's worth talking about, I think. Are there some superheroes that are possible? Well, to some extent, maybe, probably not likely, but possible. Increased healing time, for example, like Wolverine in X-Men. An increased healing time could be possible because healing time is generally coded in the genetics. So it would be possible, it would be difficult, but possible. The absorption or changing of genetic traits like that of Rogue or Toad or Mystique, or better yet, let's talk about the Hulk. Would it be possible to have an offspring that was green and had huge muscle mass? It's possible, yeah. I mean, our skin tone and our optimal muscle mass is all determined by our genetics. However, would it be possible to turn it on or turn it off if you get angry? Probably not. You don't have the ability to change your genetics just by thinking about it. Genetic markers are there or they're not. They're either expressed or they're not. They're not something you can turn on and off. Another absorption of traits would be that of Spider-Man, being able to absorb genetic material from spiders. I don't know if a Spider-Man is possible, but I know that a spider goat is, because it's already happened. Some scientists in England have hybrid or have directly mutated some goats with spider DNA, and the goats now produce spider thread in their milk, and then the scientists can then take the milk, refine it, and get the spider thread out. Why would you want to do that? Well, spider thread is the strongest natural fiber that we know of. So it's incredibly beneficial. So could that be done with a human? Possibly. It's possible. Increased strength, like the Hulk or like Beast, certainly possible. Strength can be coded in the DNA. Increased lifespan, like the Immortal or like Dorian Gray or McLeod or whatever. There's potential there. DNA at the end has little things called telomeres, and it's basically twist ties that kind of put a cap on DNA. Scientists have noted that the longer the telomere, the longer the natural lifespan. That doesn't mean you're immune to getting hit by a bus, but it does mean that generally speaking, that people with longer telomeres live longer. It also explains the fact that if you have grandparents and great-grandparents who lived till they were 90, then members of the family tend to live about the same length of time because it's coded in the DNA. Now, controlling the weather, laser vision, telepathy, telekinesis, those kinds of things, I don't think so. Those really don't have anything to do with genetics, so I don't see those happening anytime soon. But it doesn't mean that comic books will waste your time, because some of this stuff is possible, maybe not likely, but possible. So let's look at the objectives. The process of DNA replication. First of all, DNA helicase unzips the molecule. DNA polymerase copies the DNA by placing new nucleotides on the replication fork in a 5' to 3' direction. There's a leading strand, and it just replicates right on, but the lagging strand, since it's running actually 3' to 5', has to be done in little segments like a typewriter. The importance of complementary base pairing is that when DNA polymerase is copying, when it runs into an A on the leading strand, it's going to put a T. When it runs into a C, it's going to put a G, and vice versa. Same on the lagging strand. The coding ATCG is what makes replication possible. It's because it's semi-conservative. We have the original strand, and then we have the new strand. The new strand forms because of complementary base pairing. And what can happen if there's an error in translation? Well, it's called a mutation, and many things can happen, from cancer and death to beneficial characteristics like not having plaque build up or super strength or turning green, whatever. And for the most part, mutations go unnoticed because they simply don't change anything that great. And there you have it. It's a look at DNA replication and how it happens. Our next podcast will be on protein synthesis, transcription, and translation. So I look forward to seeing you then. Until then, enjoy.